I am Antonella Amicucci, a researcher in molecular biology at Urbino University. We are at Scientific Campus Mattei, where there are our laboratories. We are going to talk about truffles and our research can help to preserve its ecology, cultural and ecological value. Truffles are highly prized mycorrhizal fungi that belong to the genus tuber and that forms a fruit body, a reproductive organ, after the accomplishment of a complex life cycle during which spores that are inside the truffle germinate, giving rise to a huge filamentous network. In the soil, the filaments of opposite sex meet and merge, making possible the establishment of a symbiosis with the root plants. After that, a fruit body will develop. This relationship among truffle and root plants is a mutualistic symbiosis in which truffles that cannot photosynthesize acquires sugars from the plant, while the plant obtains water and nutrients. This life cycle is hugely influenced by several abiotic and biotic factors, such as the plant species, the soil component, the soil microorganisms, and also small animals that feed on them and enable the dispersion of the spores. The main truffles known for their delicious aromatic bouquet are Tuber magnatum and Tuber melanosporum. Tuber magnatum is widespread all along the Italian Apennine ridge, up to Basilicata and some areas of Sicily. They are also collected in Istria and in certain areas not officially mapped in Eastern Europe. Tuber melanosporum has a wider habitat and can be rather easily cultivated. Truffle has an economic, cultural and commercial value. In fact, the truffle mycorrhizal forest can be considered a heritage both for the biodiversity of the forest itself and for the benefits for human communities that lives in the areas where they grow. Research has to help to preserve this heritage, trying to find answers to solve several problems that are encountered in this field. Some of the main critical issues are uncertainties in identification, the decline of production due mainly by climate change, anthropization, deforestation, and wrong management strategies of truffle grounds. Another issue is critical issue is the assessment of truffle quality. Urbino and L'Aquila University performed research by more than 30 years in collaboration with the national and international network. One interesting research performed in these years regarded the identification. In fact, truffles are usually and traditionally identified analyzing morphological traits, specific morphological traits by expert mycologists. But unfortunately, sometimes this identification is not possible because these peculiar traits may be lost. during life cycle. For example, unripe tuber magnatum fruit body does not contain spores, so they can be confused with less priced truffles, such as whitish truffles, tuber borchi, tuber maculatum, and other species. And moreover, there is also another hypogynous mushrooms, Coiromices meandriformis, that is collected in North Africa. So they can have commercial fruits. Moreover, during the vegetative growth in the soil, the characteristic necessary for identification are completely absent. So our research groups detected specific DNA fragments specific for the different tuber species, and we set up experiments in PCR, in vitro amplification, to be used in identification the different species in all phases of life cycle. Another aspect of the research regarded aroma. Aromatic bouquet is one of the main characteristics that make truffles so appreciated. But truffles do not produce aromas for human enjoyment. These substances that are volatile organic compounds are produced by truffles for a fundamental biological role. In fact, they can be considered a sort of communication tool, useful for truffles to have a sort of conversation with the plant root and the bacteria present in the soil. Also, the hyphae of opposite sex, before merging, they have to recognize themselves, and these molecules can be useful for this recognition. In Urbino University, we analyzed the typical aromatic profile using chromatography, and we detected specific aromatic profiles for several species. Moreover, these aromatic substances are influenced by the pedoclimatic conditions. So we were able to understand the geographic origin of samples of the same species collected in different Italian regions. Together with L'Aquila and Bologna University, another kind of experiment was performed in order to understand the role of small animals. In fact, through atomic force microscopy, it was possible to observe that the passage of truffle spores through the digestive system of small animals, such as invertebrates, snails, and mouses or wild pigs. During this passage, the spore wall thins, and so it can easier germinate and easier mycorrhize root plants. Another aspect of the research regarded bacteria. In fact, a huge bacterial community is present in the soil surrounding the mycorrhiza plants, inside the fruit body, and in all phases of truffle life cycle. They have a role in the aromatic bouquet composition. They can enhance hyphal growth, mycorrhization, and spore germination. In our laboratory, we detected a Bradyrhizobium bacterium inside the tuber magnatum fruit body. This is very interesting because Bradyrhizobium has a nitrogen fixation. This means that the fungus and the plant, thanks to this bacterium, can absorb nitrogen because this element is present in the soil in a not bioavailable form, and this bacteria can fix it, making it bioavailable for the fungus and the plant. Moreover, different strains of Bradyrhizobium were also found in tuber magnatum mycelial cultivations, and these mycelia can't grow without Bradyrhizobium. This can be the starting point to obtain pure tuber magnatum cultivations to use to obtain better quality mycorrhiza plants. Moreover, different strains of Pseudomonas bacteria were used to improve mycorrhization of tuber melanosporum plantlets. In fact, it was observed that these bacteria can have a positive effect on the degree of mycorrhization. So the study on bacteria can be useful for the development of innovative and sustainable strategies to improve mycorrhization and also to enhance truffle production in the field. So regarding the decline of production, experiments were performed in order to understand how truffle can resist to increasing temperature. We used several strains of Tuberborki mycelium. Tuberborki is one of the most adaptable truffle that can be cultivated as mycelium in the lab. These strains were grown at increasing temperature, and we could observe that one of these strains can resist better to the increasing temperature. It can switch on mechanisms that enable it to grow better than the others. The applicability of this knowledge is interesting because it can be a guide for the choice of suitable species and suitable strains to be used in areas with peculiar pedoclimatic conditions to establish truffle cultivations. Another aspect of the research regarded heavy metal pollution that can cause environmental damage, biodiversity loss, and consequently also contamination of the food chain. We analyzed, also in this case, different strains of Tuberborki mycelia in cultures with high concentration, increasing concentrations of lead. We observed that some strains are more resistant in that they can sequester this heavy metal from the soil and store it in vacuoles inside the cell and not in the fruit body. These results can be the starting point to set up sustainable mycoremediation strategies to restore polluted environment. Regarding the critical issues about post-harvest quality of truffle, L'Aquila researchers set up methods using quantitative magnetic resonance to analyze the internal part of truffle and certify its quality, its integrity, and its freshness. In fact, truffles host a great variety of microorganisms and insect larvae that may cause internal rottenness and cavities that might compromise edibility and commercialization. Research is essential not only for expanding knowledge, but also to enhance this truffle heritage, to protect his value in economic, cultural, and ecological aspects.